I'm going to talk a little bit about the microbial companions that live on us and in us. And I'm going to start off, I don't know how many people, I don't know the general background of the audience. And I know there's kids here, so I like to start sort of the very sort of introductory course in microbiology. And so we start off with what are bacteria? And so does anybody know what bacteria are? You kids actually, if you guys are listening, which is also a 50-50 shot, maybe it's a 20-80 shot, definitely answer these questions. But are you guys familiar with bacteria? Do you know what bacteria are? Yes, right there. What are bacteria? Yeah, either he'll tell me what you say. Mhm. A cell, yes, very good. So that's excellent. So bacteria are, I think I have that, tiny single-cell organisms. You nailed it. How big are they? So single cell, how big are they? Can you see them? No, correct. They're very, 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 very small. You can only see them with a microscope, and about 1 billion of them can fit into that dot. Actually, there's probably about 1 trillion of them can fit into that dot. Where do they live? Any? In the body, yes. That's one place that they live. Are there any other places where we have bacteria? Yes, that's exactly it. Even the exact word I use. So yes, everywhere. Bacteria. So they're not only in and on the body, but they're in water, they're on surfaces, they're on the edge of that microphone right there, under your stove. They live everywhere. So they're clearly a part of our lives, both intimately and then also in the environment that we engage. So what do bacteria look like? They come in all different shapes and sizes. And so now we're looking under a microscope, so you can start to see the shapes. And we can stain them so that you can see them better. So these are round bacteria. They're called cocci. These are staphylococci. So if you ever had a staph infection or known people that have staph infections, that's what they look like. But they're also good staph bacteria. They're not all evil that live on our skin in particularly. They come in rods, and so here's a couple examples of rods. The rod-shaped are bacilli, and so people have heard of Bacillus anthracis or anthrax, but there's also a lot of good bacilli that live on us. Bacillus subtilis is one of them. And then finally, they're also shaped like spirochetes. There's also other shapes, but these are the three major ones. And then spirochetes look like corkscrews. And they're not shaped like that so they can get into bottles of wine. They're actually shaped like that so that they can twirl and use that as a propulsion mechanism to get through media or water. How many different kinds of bacteria are there? There are like 3, 5. Anybody? I think people are just shy. So there are millions of different kinds of bacteria. They're found everywhere on Earth. They're not all cartoons and stuffed animals, but they're there. Are bacteria, this is a key question. Are bacteria good or bad? This one's on the exam. Both, good. Some are bad, but many more are good. And I'm amazed that when I go and speak to groups, especially young children at elementary schools anywhere, both in Europe and the United States, that people know that, or kids know that bacteria are good and bad. Because given how, at least I was raised, I would never have thought that a good bacteria ever lived ever, in terms of like having to wash my hands 40,000 times a day. So what do the good bacteria do? So they help us digest and get nutrients from food. So the building blocks that make up our bodies contain amino acids, and the amino acids can be thought of the building blocks that make up proteins that make up a lot of the tissues in our body. And so some of those amino acids we can't make, our bodies do not make or cannot extract easily from food, and we rely on the bacteria in our digestive tract to make them or gather them for us. They also protect us from bad bacteria and protect us from illness. So they're almost an extension of the immune system. In fact, they talk to the immune system. One way that bacteria can protect from bad bacteria, good bacteria can protect from bad bacteria coming in is sort of thought of as what I like to think of as the good neighbor theory. We all live in houses or condominiums or apartments or whatnot, and we usually have lots of friends that live near, or hopefully not on, near or next to us. And if some of them move away, the question is, who's going to move in next to you? You hope it's somebody good that doesn't play the music too loud at night or doesn't keep you up or drive over your lawn. But also there's an opportunity for a bad element to move in. And so when we clear out some of the bacteria from the digestive tract, that leaves an opening for pathogens to come in. And so the good bacteria we want to keep there and try to avoid kicking them out by eating bad foods or introducing antibiotics when we really don't need antibiotics. So it's good to keep the good bacteria there to block out the bad bacteria. I mentioned that they talk to our immune system. We can grow mice, we can raise mice in the laboratory that never have seen a bacteria, sort of like the bubble boy mice. They're born sterile, they actually can grow up, but without bacteria, their immune systems are completely different, and they don't function properly. Sometimes there's hyperimmunity, you have allergies and asthma, inflammatory bowel disease, they have all sorts of problems like that. And then in other senses, they don't have enough of an immune system to protect themselves from an infection. But when you introduce bacteria, good bacteria, into these animals, then their immune system quickly fixes. And it's balanced and they're able to stave off infection and then not get too overwrought to the point that they start attacking their own bodies. The other element that's here next to the bottom, they work together to help us adjust to our environment. That's sort of an advanced fact that I'm not going to spend a lot of time on, but if you think about the last 130 years of the Industrial Revolution, the introduction of antibiotics, the introduction of processed foods, our bodies are constantly being bombarded by new chemicals and new good and bad, or things that potentially could be good and bad, but things that we weren't put on this earth, or we weren't here naturally able to deal with. And bacteria are evolving every time they divide. Every 20 minutes, one bacteria splits into two. One bacterium splits into two. And each time there are mutations that can help it selectively live in a particular live in a particular situation or not. And having trillions of them inside of our bodies helps them adapt to the elements that we're seeing all the time. And sometimes they'll break down those bad chemicals, or they'll tell our immune response not to get too excited and overactive in the presence of those agents. So they help us adjust to our environment as well. The question is, do we have all the same microbiome? I figured I'd just bring my problems to Sweden. So in this case, we have male, I think, and female. Do we have the same microbiome? Some people are shaking their heads. That's right. We do not have the same microbiome. You could think of it, so you could say, well, there's probably a lot of reasons why they don't have the same microbiome, but is it because one is male and female? Do men and women have the same microbiota? In reality, that's not the thing that differentiates us. I mean, we're all different. And so when we try to think about this, then we don't go there. But the fact is that every microbiome in every person in this room is different. And there are a lot of factors that help shape the microbiome in each of us. There's our host genetics, the genome that we have. There's the environment that we live in, the food that we eat, the water that we drink, and then the location that we live. Is it warm? Is it cold? Is it in the farm? Is it on a farm? Is it agrarian? Or is it in the city? And there's many, many more. So it's important to maintain this garden of bacteria on us. This fact is probably a little bit outdated. It's a very controversial number. People seem to get really upset when you put this up in a slide. The idea is that there are about as many bacterial cells on and in a human body as there are human cells. So you can think of yourself as bacterial as you are human. Those cells weigh about 3 to 5 pounds, about 3 kilos. But don't take antibiotics thinking you're going to go lose weight. But if you think about it, it's about the size and shape of an organ. And so a lot of us are thinking about the microbiome as a systemic organ, like blood, that coats us and lives inside of us and produces a lot of different functions that we need. And then the general observation, and so you can think of this actually as we move into the talking about what we're learning about the TEDDY children, is that the more bacteria that you have, generally the healthier you are, or the better off that side is. And that works in the digestive tract, on your skin. In your mouth is a little bit different. You want to have a good number of different bacterial species there. but there's chances for overgrowth if you don't brush your teeth, for example, or if you don't practice good hygiene. In that case, it can actually become too much, and it causes actually opportunities for biofilms to develop, and that's bad. But most body sites diversity is the thing that you're looking for. Can we see bacteria without a microscope? I'm not really going back into basic questions again, but I'm using this to transition into what we do with the TEDDY cohort. So in the end, there's a lot of bacteria in the environment and on us that you can actually streak out on what's called a petri dish. There's food, a gelatin food that's on the surface of this plate that you can see. That's just kind of sort of a brownish yellow color. But then when single bacteria land on the plate, they grow out and divide and divide and divide and divide and divide. And over 24 hours, you can see them. So it's just each one of those spots is a colony of bacteria that grew from one single organism that was on the plate, probably about 24 hours earlier. However, not a lot of there's most of the bacteria that we're interested in cannot be easily grown in the laboratory. So what can we do then? Actually, about 90% of the bacteria, 80 to 90% of the bacteria that live on and in the body are not easily grown in a laboratory. So in order to address this problem, we use special tools to be able to count and determine what organisms are there and how many of them are there. And it turns out that the technologies that we use to do that are the same technologies that were used to sequence the human genome by looking at the DNA of the bacteria. And so this sort of complicated slide is really, I can walk through it, is how we look at your stool samples, or your plasma samples, or your nasal swabs. And we're probably maybe getting into PBMCs. But the idea is that we can take a sample, be it a swab or a stool sample or anything that you can collect a sample with. We collect all the microorganisms out of that particular sample, and then we pop them open. And we get the DNA and nucleic acids out of it. And this is sort of the fingerprint for that particular organism. And then we gather them all together, each sample together, and we run them through a machine. And then after using some sophisticated computer algorithms, use that information to tell which bacteria were there and how many were there. And what we end up with is a microbial profile that we then can compare over time in a single person, and then compare that profile with a single person against others' profiles. You can compare healthy versus sick, you can compare young versus old, and bring in all the other information that you're collecting in diaries, and ask how the microbes associated with those pieces of information. So now we can talk about the information that we're collecting with the TEDDY children. So how do our commensal organisms, commensal bacteria, those that live on us and in us, associate with autoimmunity? You know, I just realized I didn't actually, I left out one term. So the collection of organisms, when we talked about bacteria being everywhere, the collection of organisms in a given niche is called a microbiome. How many here have heard of the term microbiome? Okay, maybe there's a Swedish term for it. Probably not. Probably microbiome. Yeah. So the idea is that, so the collection of bacteria in any given environment is called a microbiome. So all the bacteria on and in the human body is called the human microbiome. So if you hear me use that, I'm just referring to the collection of organisms in a specific niche. So you all are familiar with the TEDDY study, so I don't have to go into this. You know it better than I do. Some wonderful investigators that lead the study and some wonderful clinical coordinators that help make it a reality for all of us here. So as Dr. Lernmark mentioned, we are looking at all the stool samples. And you're thinking, what a great job that is. But it actually, I mean, from a science standpoint and reality, it really is, because the TEDDY study is unprecedented, not only for type 1 diabetes, but in child development from birth. And I'm going to reiterate that point as we go through, because you'll see some of the remarkable results that we're seeing that nobody else is able to see with the context of health or disease. So the TEDDY cohort can be broken down into two groups. What we call the first islet autoimmunity or first autoantibody group. That is where the children who have had the first appearance of an antibody, one single antibody. And then there's a nested case control group for type 1 diabetes. So the outcome being you have type 1 diabetes. They each have a matched control, matched by clinical site, so where you're located, whether you're male or female, and whether your first-degree relative is also at risk. We take these two cohorts and all the samples within them, so there's 9,747 stool samples associated with the autoimmunity cohort, and 3,508 associated with the type 1 diabetes cohort, and we're looking at everything. We're looking at the bacteria, we're looking at the fungi, we're looking at the viruses, and all these samples and associating with clinical outcome. Of course, we have all this wonderful metadata, the diary information that we collect, the other clinical data that are collected to compare with it. And then we can ask some very interesting questions. And so the first question here is, and this doesn't necessarily show up, but I think we can get enough out of this. So if we're looking on the right, so this side over here, what we're looking at is the development of the microbiome over time. So time in months is down here, what we call the x-axis. And then these numbers here, just thinking of it as the change in the microbiome from point to point. So if I'm taking a sample in January and comparing it to a sample in February, how much has that sample changed during those 2 periods of time? And then going on. And then we can group the samples by whether they came from somebody who's autoantibody positive or who is the control. So autoantibody positive is red, control is in blue. So we'll just follow the blue curve for a second. For the first 10 months, we can see that there's sharp changes happening in the microbiome. So the first 10 months of life are really the most important in terms of assembly of your microbial you. You're born almost sterile or basically sterile, and then the first contact you have with your mom and your father and the hospital, and then eventually your home, those organisms work their way inside of you and you collect this community of organisms. About 10 months, so anywhere from 10 months to 2 years, you stabilize. They're all there and you change a little bit over time, but it's constant. New organisms come in, some ones die off, sometimes they come back, and so it's stable after 1 to 2 years of life. You contrast the blue curve to the red curve, and you can see that there's this still up and down pattern moving forward away after 10 months. That's different from the controls. And so what we're looking at there, in my mind, is an attempt of the microbiome, the collection of bacteria, to gather together, but then as they get to a certain critical threshold, the community collapses, and it starts to build up again, and it collapses. And so we think of this as an instability. And the only reason we can see this is because we have all of these samples these first 35 months. If we only took one sample a year, we'd have a point here, 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 and here. And that wouldn't be enough information to see this instability. And no cohort with this size and this depth has this type of data to be able to discern this observation. So this is a first in the microbiome field, especially in humans. So we built this hypothesis, based on an initial observation, that islet autoimmunity is associated with the failure to reach and maintain this intestinal microbiome maturity in the first few years of life. So the next thing that we did is that we wanted to take a look, all right, how does every sample compare to one another? So every dot on this spinning plot represents one sample. And the distance between any two dots in this plot is exactly proportional to how identical those two samples are. So essentially, any two dots that are close together, the organisms that are in them and their relative abundances are very similar. The further apart they are, the more different that sample is. They have different bacteria, they may have different distributions. And then we can label them based on different types of information that we know. So in this case, the red samples are from cases and the blue samples are controls. One of the observations that we could have found here is that all the red samples over here, all the blue samples over here, but they're not. They're all intermingled, which suggests that the bacteria in general that are found in the children of our cases and those that are controls are the same, more or less, at different points in life. So nothing really stands out at this sort of high-level observation. If I change the labels to become matched by age, we can see that, just like we talked about, the microbiome changes over time dramatically in the first few years of life. So red is 2 months of age, and blue, dark blue is 72 months, and then the shading is everything in between. You can see on the left in particular that, first of all, we have two clusters that jump out at us. One is about half and half blue and red samples, but the blue all cluster to one side and the red all to the other. And then the other cluster is almost 99% red. So a lot of young samples in here, a lot of older samples in here. Again, this sort of distribution of samples, this two bigger clusters, not seen before. And again, it's because of the discrete sampling. It's because you all have taken the time to bring in stool samples and help make this type of observation real. You're like, well, that's great. You have 2 spots on a slide, and I'm collecting poop at home. So give me a moment. So we have this instability hypothesis that we wanted to test. And so now we have those same 2 clusters I showed on the last slide. They're now colored red and blue. And thinking about the fact that we have 20 to 50 samples per child, we can track their progress through time. And in a way, we can measure how often they're in the red cluster and how often they're in the blue cluster. And so I depict a cartoon at the top left to sort of show what's happening. The young samples, as we know, start off in the red cluster. And then the next sample that's collected, half the time moves into the blue cluster. So January to February. January you're red, February you're blue. Or half the time, the next sample turns out to stay in the same red cluster. Once you're in the blue cluster, 90% of the time, your next sample stays in the blue cluster. But 10%, you see the 767, they jump back to the red cluster. So, all right, so that sort of stands out to me, is that if we're going through a natural progression through assembly of the microbiome, shouldn't we keep going in that direction? The fact that some are going back suggests, again, I think about sort of the house of cards model, where cards are being stacked up and then you get to a certain point and then there's a collapse. You build them up again and there's a collapse. So is this a signature of that event? So the next thing that we did to look at this even further is we took a case and a control and we started comparing their samples moving through the clusters over time. So the clusters I'm holding static, just to make it easier to see these 2 children's samples over time. Time is ticking away in months up here. And you can see that one sample comes from the red cluster, moves to the blue cluster, and stays, whereas this child's samples is bouncing back between the 2 clusters, reflecting instability. So this turns out to be the case, the one that's bouncing back and forth, and the one that jumps and stays is from a control. So that, to me, it's again, an observation. Making this type of observation is only made possible, again, by the discrete sampling that TEDDY has done, and that you all have done a great job of contributing to. So the next question we had, and you heard a little bit from Dr. Lundmark's talk earlier, is this observation widely distributed across all children? Is it specific to certain antibodies? So if the first antibody happens to be GAD, for example, do those children jump back and forth more frequently than, say, IAA? And so we asked that question, and now you have this bingo ball observation. But we colored the samples by whether that child had 2 autoantibodies that appeared at the first visit, it was GAD only or MIAA only, or if it was a controls in black. As you can see, everybody's jumping across. So it doesn't seem to be anything from this perspective associated with the first autoantibody appearance. Again, time is ticking away at the top, so we're about 13 months right there. You can see that the balls are slowing down. Again, thinking about the fact that you become, generally you become more stable after about a year of life is reflected now in how fast the spheres are moving. They're slowing down. You're not changing as much. And it's going to turn over in a few seconds, and you can see right after you're born just how fast things are changing in those first few years of life. You don't have to be worried about, you don't have to be thinking about type 1 diabetes when you look at this. This is actually the first sort of image of how the microbiome changes over time in children, period. Western Europe and the United States. There's no other data set like it, and it really lets us look at the speed of change in the gut community, which is now starting to reveal potential characteristics that may associate with disease. So to that end, we have this going on. So how do we measure this in a way that we can make some sense of it? We can bring statistics into this. Because showing movies and papers is not going to get us any further in solving type 1 diabetes. So in this plot, what we did is we wanted to create some sort of some sort of community descriptions that we can categorize each sample, where we can categorize each sample. So to say it more clearly, using a certain set of software, we can look at each sample as belonging to one of 6 different community state types, or CSTs. So if you look at these, there's this group here, 1, 2, there's 3, there's 4, there's 5, and 6. Each one of these is a community state type. And while it's not easy to see how they're distinguished down here. If you look up here, this light blue suggests that these organisms are highly abundant in each of these samples. So, for example, group 1 here is dominated by this organism called Faecalibacteria prosnitzii. This group 2 is Rhumnococcus bromi, or breve. Whoops. This is Bifidobacterium longum, and so on. So we can actually identify any sample that we've collected and pin it to one of these 6 categories. So, well, that's good. What are you going to do with that? So the next thing, well, first of all, we wanted to ask, does any of that associate with being a case or control? Does being a state 6 mean that you're highly likely to get to type 1 diabetes? And looking up here, we labeled each sample in blue by control, in red by case. And you can see that all the community states have red and blue in them. There's really no statistical significance associated with the community state types and outcome. Another way to look at it, though, so we still have our community state types now represented as spheres. The size of the sphere is proportional to the number of samples that fall in that sphere, so that state type 1 has the most samples associated with it, and state type 6 has the least. Again, when we color them by whether they're a case or control, the middle part is black or blue, which suggests that all of these, since they're all blue, they're about an equal chance of being a case or control. But all these arrows, just to simplify, is the fact that we can measure the transitions between each of the states. And so when we do that, we actually learn something really interesting. We find out that the number of community states that you see, the number of different community states that one child's set of samples falls into, turns out to be associated with a higher odds risk for becoming autoantibody positive. So, and now we can assign statistics to it because we have an actual number and we have standard deviations. So, and what these numbers on the left suggest is this is how many days before the first autoantibody was detected that we saw a significantly different change in the number of states before that antibody was detected. So in this case, if you read this top part here, this means that 400 to 500 days before the first autoantibody was detected, we saw that the number of community states in children were higher if it was going to be diagnosed than those that compared to their control. 300 to 400 days out, 0 to 100, basically looking at all samples, there's a statistically significant difference in the number of community states that a child's gut microbiome goes through if they're going to become autoantibody positive. So there's more instability. We can now measure it and then give it a weight to it that we can then move forward with and try to understand why. We also found out that it's not the number of states that you go through, but also the number of could also be the number of transitions. Same story, 400 to 500 days out. A year and a half before the first autoantibody is detected in the lab, we're able to see changes in the gut microbiome being associated with onset. And so what does this mean? So if you were going to, so how can we translate this into something useful for practice and caring for type 1 diabetes? So looking back at this slide, you can think about, collecting those stool samples. So say you're collecting the stool samples still monthly and you send them into a clinic. That clinic does the same type of profiling we do. It's kind of unwieldy for a diagnostic, really, sequencing all these samples and then analyzing them, although it's getting easier these days. But if you go back to these 6 organisms, we can create assays for these 6 organisms pretty easily. And then when your sample comes in each month, we can go quickly check for the presence of these 6 organisms, and if one of them jumps out as more abundant than the others, we can say, oh, look, there's a lot of Bifidobacterium longum in Stacy's sample. She's in state 3. We can do this month by month and then count and assess the transitions that are there. So does Stacy a 3, 3, 3, 3, 3? Probably okay. But if Stacy's going 3, 4, 5, 4, 3, 2, 3, then you have a chance that there's a problem, or at least an increased odds risk for becoming autoantibody positive. Or if it's 3, 2, 3, 2, 3, 2, 3, 2, same type of thing. Number of transitions being associated with onset. This has to be validated. This is an early observation, but again, made possible by the fact that the TEDDY program and you all are collecting those stool samples. So we're very optimistic that we can hopefully turn this into something that could be useful in the clinic. That doesn't speak to causality. So right now we're still talking about diagnostic. What can it mean, though, in terms of potentially identifying sources, the cause of type 1 diabetes? Problem with the microbiome field, or with all science, areas of science really, is that when you have an observation, you have to determine if it's the chicken or the egg. Is it a result of something happening? Is it an environmental exposure? Is it the instability of the immune response that's being reflected in the bacteria in the gut? We don't know. So these are experiments that we're going to have to do moving forward. And then if we do identify those, perhaps those things become actionable, that we can become thinking about targets for therapy. And so with that, I'm just rehashing everything I just told you. Thank you for your attention, and I'm happy to take any questions that you have. And Oka can translate.